Alrighty, hello everyone. Welcome again to your first discussion recording. I tried to post this last night after multiple attempts, and I got a few emails from y'all today saying that the screen froze after the first question. When I went back and reviewed the video, that was exactly what happened. So this is attempt number 6 to go over discussion number 1. So hopefully everything works out this time. So I'm going to go ahead and flip over to my iPad and we will hop on into it. So let me get that going and we will begin. Alrighty, iPad screen share, here we go. All right, awesome. So this is discussion exercise number 1. Looking at question number 1A, what we're going to be doing is looking at the different ratios of the concentrations of different molecules inside and outside of the cell. So whether they're in the extracellular fluid or the intracellular fluid. In this example, we can say that this is a muscle cell because it is not specified. If it's not specified, we can say it is any type of cell that we want it to be. Muscle cells will be the same as neurons, renal cells, kidney cells, all those other cells except for epithelial cells. And I'll discuss that as we get further into this video. But muscle cells and all other cells except for epithelial cells will be alike in everything we are about to label. So if you see here, I have already put down sodium, potassium, calcium, glucose, oxygen, and carbon dioxide. That's what the question is asking for. If you notice, I did not put H2O down, water, because that will be kind of covered in our next discussion when we get into osmosis and tonicity. But let's go ahead and begin. So sodium will always be in higher concentrations outside of the cell and lower concentrations inside of the cell. So sodium is always going to be higher outside and lower inside. Sodium and potassium are going to be inverse of each other. So they're always going to be opposite, meaning that sodium will be lower outside of the cell and higher inside of the cell. Now, calcium is just like sodium by being lower inside of the cell and higher outside. Glucose, however, is a little bit different, especially when it comes to our muscle cells and all of those other cells. And what we see is that glucose is going to be in higher concentrations outside of the cell and lower concentrations inside of the cell. And you might be wondering, don't we need glucose for, in this example, like muscle contraction? Don't we need to break down glucose to form ATP? And if you are thinking that, you are correct. And I can kind of explain to you why we see this higher concentration outside instead of inside of the cell. If you remember back to functional biology, your intro biology course, the first thing that happens when glucose enters the cell is it undergoes glycolysis. And glycolysis is going to phosphorylize that glucose. Anytime we talk about phosphorylation, that's going to mean adding a phosphate to that glucose, and that will lock it into the cell. So the first step, moving that glucose into the cell, is going to be glycolysis, and glycolysis will transform that glucose into glucose 6-phosphate. Glucose 6-phosphate, and that's going to mean it is phosphorylized and locked into that cell. It's important to know that glucose and glucose 6-phosphate are not the same thing. Therefore, we're going to have lower concentrations of glucose inside of that cell and higher concentrations of glucose 6-phosphate, which is what we can actually use to break down to form some ATP. So lastly, we have our two gases, oxygen and carbon dioxide. Oxygen is going to be in lower concentrations inside the cell and higher concentrations outside. And carbon dioxide, again, will be inverse. It'll be higher concentrations inside because our cells will go through metabolism, and the byproduct of metabolism is carbon dioxide. So we'll have higher concentrations of carbon dioxide inside the cell and lower concentrations outside. So with this knowledge, we can kind of look at the next question: which molecules can move by simple diffusion? Simple diffusion is a form of passive transport. There's no membrane protein required, no ATP required, and the molecules that we're going to see that can move by simple diffusion are going to be our gases. Our gases are very small, nonpolar, non-charged molecules, so they can simply pass that membrane without any protein channel or energy required. So our two molecules that can move by simple diffusion are going to be our O2 and our CO2, our gases. The next one asks which requires some type of transporter. Well, it's going to be the rest of them, our ions and our glucose. So our ions and our glucose. So before hopping into question number 2, I kind of wanted to discuss the difference between a channel and a carrier. So what we see is, I'll go ahead and draw out a diagram to kind of explain the difference between these two. And if you're drawing along with me, I would go ahead and watch me draw this out first, because I will be erasing on my iPad some portions of it. And I know how frustrating it can be when you're drawing with an actual pen and then the professor erases something and you're like, oh, I can't erase that with a pen. So if you're drawing with a pen, following along with me, maybe watch me draw this out and then redraw it once I am finished. So a channel is a type of membrane protein that will create a hole in the membrane and open it up for some type of ion to move through it. So it'll create a hole and open up that membrane for some type of ion to move through it. So for example, it would look something like this. Let's say we wanted to move a molecule of sodium into our cell. We would place a channel on that membrane, and that sodium could simply move right on through into that cell, into that extracellular fluid from that intracellular fluid. However, carriers are different. They're different because they only open to one side of the cell. So they're only going to open towards the extracellular fluid or the intracellular fluid. So our carrier channel, or our carrier transporters, are going to look something like this. They will kind of open up. Let's say they're grabbing a molecule of glucose. That glucose will bind to that carrier, and that carrier will go ahead and once that glucose is binded, flip over and let that glucose enter the cell. So carriers have a very specific binding site, and that they only open to one side of the cell. Channels do not have a specific binding site. They can just open and let some type of ion travel on through. All righty, nice. So moving on to question number 2, now we're going to look at some of these specific molecules. We're going to look at some type of movement, why they move that way, and how they move that way. So we see that question, or column number 2, is looking at the means of transport. So whether it's moving by simple diffusion, like requiring no energy, it's passive, whether they're requiring some type of active transport, which needs energy, etc. And then column number 3 is talking about why it's using this means of transport. So why we're using simple diffusion, or why we're using facilitated diffusion. So looking at the first row, we're looking at the movement of sodium from the extracellular fluid into the cell. So moving sodium from the outside of the cell to the inside of the cell. And sodium, as we saw before, is going to move down its concentration gradient. So it's going to move from an area of high concentration to an area of low concentration when entering a cell. This is a form of passive transport because it does not require energy to move across or with its concentration gradient from high to low. However, sodium is a charged molecule. It has that plus, that positive sign next to it, Na+. That means it is a charged molecule. And charged molecules cannot move by simple diffusion. They're going to need some type of help. So what we see is that sodium is going to move from the extracellular fluid into the cell by simple diffusion and also by some type of movement through a water-filled channel. We're going to need to open that sodium channel, as I drew up here, to let that charged molecule inside. Now, if you're looking at this and you're like, what is a water-filled channel? I can kind of explain to you what that means. So going back up to this example, and oops, I see that I messed up that intracellular and extracellular, so sorry about that. I kind of flipped them over. This should be the extracellular and this should be the intracellular. But in that extracellular fluid, there's always going to be a lot of water molecules. There's tons of water molecules. They're very prevalent in all areas of our body, especially in that extracellular fluid. And anytime we open up a channel, whether it's a sodium channel or any other type of ion channel, water is also going to move through, because water will naturally move through any channel. All channels are water-filled channels because water is going to move through any type of channel. Hence why we normally just call them channels. Specific channels for water are called aquaporins. However, water will move through all other types of channels. That's why we normally just call them channels. So don't let this water-filled portion confuse you. We're just talking about moving through our channel. So column number 3 is asking about why, and we discussed that earlier as well. It is an ion. It is moving down its concentration gradient. And one thing we didn't discuss is that it is also following its electrochemical gradient. So it's following its electrochemical gradient. This is something that we're not going to get too deep into during this class, but I will quickly explain it to you. It makes sense once I draw it out. And yeah. So when we're talking about our electrochemical gradient, I'll use sodium as an example. So we know that sodium is in higher concentrations outside of the cell. So we have lots of sodium ions kind of floating around outside of the cell. And if you remember, sodium ions are charged. They are charged. They are positively charged. Positive charges repel each other. Remember, opposites attract in science. So positive and negative attract. However, like charges repel each other. So if a bunch of sodium ions are positively charged right next to each other, they're going to repel off of each other. They're not going to want to be near each other. And this will kind of cause this electric repulsion, this electrostatic repulsion, and kind of drive those sodium ions into the cell. So they'll kind of repel off of each other, causing this repulsion of them, kind of forcing them into the cell even more. However, we're not going to get too deep into that, that mechanism of the electrochemical gradient. That's just what that means in case you were wondering. All right, nice. So our next example that we're going to talk about is aldosterone moving into a target cell. So aldosterone is one of our steroid hormones. It's a pretty small non-polar molecule. We're going to learn a lot more about it in future lectures. You aren't expected to know what it is now, but aldosterone is going to move into some type of target cell. But again, it is a small non-polar molecule. So what we see is that aldosterone is going to move via, sorry, give me just a second here. My iPad is lagging. Sorry about that. Back to the question. But again, aldosterone is one of those small non-polar molecules. And whenever we have a small non-polar molecule, it can easily move by simple diffusion. It's small, it can easily move past that lipid bilayer, and it's also nonpolar, so it's not charged. It can move through easily. So we'll see that aldosterone is going to move by simple diffusion because it is a nonpolar molecule and it is moving down its concentration gradient. So our next two are looking at glucose moving into a renal proximal tubule cell and glucose moving into a muscle cell. And I actually am going to use a diagram to explain this to y'all. So if you want to take a second, and let me add one thing to this. If you want to take a second and pause this video, to draw this out, I'm going to add a lot of extra notes to this, but this is the basic layout. So if you want to take a second, pause and draw this out, go ahead and do so, and welcome back. All right, awesome. So before we get too far into this, I just want to make a quick clarification. We haven't learned exactly what those renal proximal tubule cells are. So for now, I just want you to think of them as epithelial cells. They are a type of epithelial cell. So for now, that's just what we're going to think of those renal proximal tubule cells as. So let's go ahead and move on down to the diagram. So what we're going to kind of be looking at here are our end goal is going to be getting a molecule of glucose into the muscle cell. So our overall goal is going to be getting a molecule of glucose and moving it on into the muscle cell. However, we can't just do that. There's a couple steps that we need to do before. So let's go ahead and discuss those. So glucose, once we ingest it, it's going to enter into our esophagus, move into our stomach and all of that. And it's going to enter the lumen of our kidney or our small intestines, things like that. So anytime we say lumen, what we're meaning is the outside of the body. It has not yet been absorbed by the body. Therefore, we can't use it for metabolism. We can't use it for energy production. It has no use when it's inside of the lumen. If it's not absorbed, it'll be excreted through the urine. If glucose is being excreted through the urine, that's a sign of diabetes. So we really want to get these important molecules and ions out of the lumen and into our cells of our body. I'm sorry, my cat, if you watched my introduction, I have two cats. They love to get my attention. Right now, I don't know if you can see, but my cat is sitting on top of there, acting like she's gonna jump off, so I'd pay attention to her. But I know she's a pussy cat, so I know she's not gonna do that, LOL. But all right, back to the question. So in this example, we're going to use glucose, as I said earlier, and we're also going to use sodium. And the way I'm color coding this is either in orange will be relative to our epithelial cell, and blue is going to be relative to the muscle cell. So everything I write out in orange right now is going to be relative to the epithelial cell. So here we can draw sodium across. Let's see, sodium. And relative to the epithelial cell, we know that sodium is always going to be in higher concentrations outside of the cell and lower concentrations inside of the cell. Higher concentration outside. Sodium will always be like that. However, glucose is unique to epithelial cells in that it's higher concentrated inside of epithelial cells. In all other cells, glucose is going to be in lower concentrations except for epithelial cells. Epithelial cells have very large concentrations of glucose, meaning that the outside will have lower concentrations of glucose. So if you want to go ahead and draw that along with me. So again, our end goal is to get this glucose molecule into this muscle cell, but to do that, we need to absorb it into that epithelial tissue so it can be inserted into the blood, and that blood can deliver it to the muscle cell. So to do that, we're going to use 4 different mechanisms. So those 4 different mechanisms are all types of channels. So we're going to use 4 different channels to do this. We're going to use a sodium channel. We're going to use a sodium potassium symport. We will use a sodium sure I get this one right. Sodium, potassium, ATP ACE, and a GLUT carrier, And in the next discussion video, the part 2 of this video, which I'll be sending out by some point by the end of the night, we'll be discussing these 4 channels a little bit more in depth. And I'm sorry, GLUT carrier is obviously not a channel. The first 3 are channels, GLUT is the carrier. But these are the 4 different mechanisms that we're going to use to get that glucose into that muscle cell. So getting that glucose from the outside of the body to the inside to the specific location that it is needed. So first step is going to be moving that glucose from the lumen of the kidney to the inside of that epithelial cell. So moving it from the lumen of the the lumen of the kidney into that intracellular fluid. But what we see when looking at the concentration gradient is that glucose is lower outside and higher inside. So that means that it's going to need to move against its concentration gradient. It needs to move from low to high, which is not moving with its concentration gradient. That's moving against it. And to do that, we need some type of energy. And so what we'll see is that sodium will kind of assist it. Sodium is going to come in over here. And when glucose needs to enter into the cell, we're going to open a channel that sodium will also move through. And this is going to be our sodium potassium symport. What we see is that I am so sorry. I made a mistake when labeling these. It is not our sodium potassium symport. It is our sodium glucose symport. And what we see is that sodium is moving with its concentration gradient. It's moving from high to low. And when it does so, it's going to give off some energy to that glucose so that glucose can move against its concentration gradient. So when that sodium moves with its concentration gradient, it's going to give off energy to that glucose molecule to move against its concentration gradient. This is a form of secondary active transport because that sodium is giving energy to that glucose to move against its concentration gradient. I said that 800 times just now. Hopefully y'all got the point. But again, that's a form of secondary active transport. That sodium is moving with its concentration gradient. That gives off ATP to glucose to move against its concentration gradient. Diet Coke break. Addicted to Diet Coke. I will probably have one with me every single hot day. Once it gets cold, I'll have a coffee with me. But yeah. All right. So another way that sodium can move into the cell is just simply through a sodium channel. We looked at that above, right in here, just simply creating a hole inside of that membrane, and that sodium will simply just move on through. So now we have our sodium and our glucose inside of the cell. And now we need to get it out of that epithelial cell into the extracellular fluid, the blood, to get it to that muscle cell. So when looking at glucose, we see that it can easily move with its concentration gradient. It's going to be moving from an area of high to low. So glucose can move through that channel from an area of high to low concentration via facilitated diffusion, just moving with its concentration gradient. However, sodium is now going to be needing some type of energy, some type of ATP, because it's going to be moving against its concentration gradient from an area of low concentration to an area of high concentration. So what we're going to use to do this is we're going to use the sodium potassium ATPase. So whenever sodium enters into the extracellular fluid, potassium will move in the opposite direction. So whenever sodium enters, potassium is going to move in the opposite direction. When it does that, it's going to give off some ATP so that sodium can move against its concentration gradient. So that is that sodium potassium ATPase. All right. So everything up until this point has been relative to the epithelial cell. Now I'm going to move colors because we're going to be talking about things in terms of the muscle cell. So everything is going to be relative to the muscle cell now. So we have our sodium, we have our glucose. We can label that in the muscle cell as well. And again, sodium is always in higher concentrations outside of the cell, lower concentration inside, and glucose is always going to be lower concentration inside, higher concentrations outside when we're referring to any type of cell other than a muscle or other than an epithelial cell. So remember, glucose is unique to epithelial cells and that it's higher inside. Every other type of cell, muscle cells, neural or neurons, renal cells, all those other cells are similar in how glucose is concentrated, except for that epithelial cell. So what we're going to see is that glucose, we can begin with sodium. Sodium will just move by simple diffusion with its concentration gradient from an area of high concentration to an area of low concentration. Remember, it is a charged molecule, so we'll simply use just another one of those sodium channels to move it on through, just creating a hole in the membrane. But glucose is going to need to move glucose will also be moving with its concentration gradient. So we will use facilitated diffusion as well. And the specific type of facilitated diffusion mechanism we'll be using is that GLUT carrier. So that GLUT carrier. And again, that GLUT carrier is going to carry out that facilitated diffusion. All right, nice. So with this information, we can now kind of begin to dissect these last two questions. So what we see is that whenever glucose is moving into that renal proximal tubule cell or that epithelial cell, it's going to be moving by active transport. We had to actively move that glucose right here into that epithelial cell by this active transport, that's sodium glucose symport. Why it does this is because glucose is an ion. So glucose is an ion. We cannot simply just move it into the cell. Whenever we look at the next question, moving glucose into the muscle cell, we will do this by facilitated diffusion. So this is looking at glucose moving into that muscle cell. That is, again, facilitated diffusion by that GLUT carrier. And why we do this is because glucose is moving down its concentration gradient. So it's moving from an area of high concentration to an area of low concentration. So that is it. That is all I have for y'all for part 1 of this video. Part 2, we're going to knock out these last few questions and I'll be sending that out soon. But in case you're hopping on studying real early before the weekend starts, I wanted to get this to y'all in a timely manner. I'm sorry about the last couple videos I've been trying to upload. I'm new to recording lectures. I've had to get a couple different types of technology to do so, but hopefully this one works out, and I will see y'all for part 2 soon. All right, goodbye everyone.